Thank you to Nanopore for having me back at this wonderful conference. I'm very happy to be here. So here are my disclosures and my disclaimers. And I very briefly wanted to introduce the MGZ first, or the Medical Genetics Center. So we're an accredited human genetics laboratory based in Munich in Germany. And we perform genetic counseling at 15 different locations in Germany, but we also receive samples from all over Europe. So the company was founded in 2000 by Professor Dr. Elke Hlinske-Fedder, and we've grown a lot since then. We perform over 42,000 analyses per year, over 6,000 genetic counseling sessions. A lot of what we do is exome sequencing, but we have a strong focus on R&D and trying to introduce long-read sequencing in the clinic. And we're 200 people altogether. And we're not focused on any one area of diagnostics. We're interested in oncology, neurology, even prenatal diagnostics, and prevention diagnostics as well. So today I'm going to talk about our repeat expansion workflow. So repeat arrays are repetitive, simple nucleotide regions. So every one of us have repeat expansions in us. So it's things like CAG over and over and over. But when they reach a certain length, they can become pathogenic, right? And there are over 40 repeat expansion disorders known, mainly involved in neurological impairment. And the disease severity and the age of onset is correlated with the repeat length. So the longer the repeat length is, the younger a patient can develop symptoms, or the more severe the disease. And repeat arrays are unstable and can expand when inherited. So subsequent generations can have more severe diseases or earlier age of onset, and this we call anticipation. So this is just showing that repeat expansions can occur anywhere in coding regions and non-coding regions. Typically in coding regions, they're shorter, less than 100 repeat units, and in non-coding regions, they can be up to or over thousands of repeat units. And this plot here is just showing the number of repeat expansion disorders identified over time. And you can see that there's quite an increase in the long-read sequencing era. And that's because we get these long contiguous reads. We can capture the whole repeat expansion. We can look at the motifs, repeat interruptions. So this is helping us to discover new repeat expansion disorders. So in 2013, we published this paper looking at repeat expansion disorders that are involved in cerebellar ataxia. So we targeted 10 regions, and we did this with the CRISPR-Cas9 kit. So we chose this approach because the coverage is much higher. So you use CRISPR-Cas9, you design the guides for your region of interest. So we went upstream and downstream of our genes of interest. And then we enriched for these, sequenced them, and we looked at the repeat lengths, the interruptions, the methylation levels. And here is a plot of the coverage. You can see that the coverage for these genes is quite high. It can be from 100 to 1,000x coverage. We aim for 50x coverage for our diagnostic workflow. So the diagnosis of these genetic disorders, of these cerebellar ataxia disorders, it needs to be on a genetic basis because the phenotypes are quite overlapping. And this requires standardized and comprehensive testing. So with our assay, we could explain 28% of the patients that came into the clinic as having repeat expansions. This jumped up by 10% after this paper was published, actually, after the discovery of a repeat expansion in FGF14. So we're up to around 40%. And we can also look at many other things like repeat length distribution, repeat composition. In this paper, we identified a couple of novel motifs, and also methylation level, which we know comes for free with the Nanopore data. So I just want to show one case study where methylation information was important for the final diagnosis. So we had a male that came to the clinic. He was 62 years old. He presented with a slight intellectual disability. He had a progredient gait disorder for 3 years. He had cerebellar symptoms, and his MRI showed atrophy of the cerebellum and the cerebrum. So he was diagnosed with a hereditary ataxia of some kind. So we did our workflow, and we saw in this patient that he had a repeat length of 97 to 359 repeats. It's quite variable. And with this, we would have said that he is a FSX male. But then we looked at his methylation level and the promoter, and we saw it was lower. So why is this important? Because for we want to have the right diagnosis because we want to understand the pathomechanism, right? So when you have an FMR1 repeat expansion in the healthy range, you have a normal level of FMR1 expression. Then if you have between 55 to 200 repeat units, you have an increased expression. And then when that goes beyond 200 repeat units, you have methylation of the promoter. So then the gene is silenced. So in this case, we could actually diagnose him as a high-functioning FSX male with FX test. So it's a more complicated diagnosis, but having both of these pieces of information was very important in the end. So this was all well and good, and then Nanopore switched to the R10 chemistry, as we know, and the Cas9 kit was discontinued. Unfortunately for us, we have this as an accredited test in Germany, so we have to continue with it. So we thought, okay, we're going to make it work for R10. So with the help of our wonderful application specialist, Isabel, we worked on testing different parts of the library prep, like increasing the ligation, introducing a cleanup. We tested things like exonuclease cleanups to remove off-target DNA. We also redesigned guides so that they would change the orientation of the Cas9. And in the end, we came up with a protocol that managed to work a bit better than what it initially did when we just tried to take the R9 protocol for R10. Right when we first did this study, we had half of the coverage with the R10 flow cells and with R9, but I'm happy to report very recently, it's not in this presentation because it's so recent, in the last month the coverage has doubled or even tripled, so we're at the same level as the R9 results or even higher. We also switched to ONT's human variation workflow using the STR quantification tool, which has been a game changer for us. One, because it's maintained. So before we were using Strike. So this is a deprecated tool, which is not something that you want to use in the clinic. And the other aspect is that it uses basecalled data. So this means that if there are any future chemistry changes, which I can imagine might happen in the future, then we can continue to use the same bioinformatics workflow. And the nice thing about this tool is that we also get a very nice report, which our geneticists are very happy to see very nicely the results visualized. And we can also look at the repeats and the interruptions. So all of this, all the troubleshooting, all the protocol is in this preprint. We did try multiplexing. This is a question I get very often. We tested so many different ways to make this work, and it failed every time quite dramatically. So we haven't managed to get it work. I think other people are working on this, and it would be amazing to have it, but yeah, unfortunately, that's not the case. But I think it's important to show that these things are not working. And a nice aspect of the paper is that we have expanded our panel. So initially we published with 10 target regions. We're now up to 27 regions that we've validated, where we have many more in the pipeline. And when new disorders are discovered, we're also putting those into our workflow. And we've also introduced different panels for various diseases like ataxia, myopathies. So all of this is in the preprint, and I think it's something that we are doing a lot in the routine lab now, because we find a lot of patients that are coming in with hereditary ataxias, and we can look at their repeat expansions and give them a proper analysis. So this is, of course, the work of many people at the MGZ, so I want to thank them for all of their work. And if you ever want to collaborate on any clinical questions or method development, we're very open for that. So thank you.